Right. Welcome, everybody. Welcome back to another When Things Go Wrong episode webinar series presented by the GMP Collective. I'm really excited to have you guys join us. I know we took the month of April off, but we are back with an extra special episode. I am super jazzed to have folks from 2 countries here to discuss this topic, which I think is really important from a perspective, recognizing that most of our audience is typically in the US. You know, we have a federal framework where it's federally illegal for cannabis. And so we have this, very disjointed and fragmented state marketplace. However, the reality is that I think you're going to find today there's actually a lot of similarities, despite the fact that, Canada has a fully legal framework, has a federal oversight with Health Canada, whereas the states do not. So we are going to jump right in to our discussion around lab testing. Could your results be wrong? We've got laboratory representatives from Oregon, Florida, and British Columbia. We're going to look at several issues of lab errors that are common to the cannabis industry. Again, as I mentioned, not just in the US. And what I found is that a lot of folks, there's been a lot of attempts at solutions in this marketplace, but obviously if we had the solution, we wouldn't be here talking about it, especially at least 6 years since some of the first peer-reviewed articles came out highlighting the magnitude of these issues. So I'm really excited to be joined here today, and I will turn it over to our panelists to introduce themselves, and we will get kicked off. As a reminder, this is an interactive and engaging webinar, so I've got a good amount of questions that came in during the registration process. We will try to get to as many of those as possible. But if you've got a question or a perspective to share in a comment, throw it in use the Q&A session to throw in a question or comment, and we will do our best to answer it live for you. This is a great opportunity. So put our panelists on the hot seat. And with that, I would like to kick it off with Jini Glaros, who is here from Florida. So great to have you, Jini. Give a brief introduction of yourself for the audience.
Hi, David. Thank you. So my name is Jini Glaros. I am the Chief Scientific Officer at Modern Canna Labs. We are a Florida third-party testing laboratory. So we test medical marijuana in Florida. It's a medical market. I've been with the company since 2017, developing our methods, our protocols, standardizing things, making sure that, we can push the industry in the right direction.
Awesome. So great to have you, Jini. And I remember we met during the pandemic. So it's been great to get to work with you 3 years in now and have you around in the circle. So thanks for being here. Rob, one of my favorite colleagues from Canada. So good to see you, sir. Introduce yourself.
Yeah, Rob O'Brien. I'm a former university prof focusing on analytical chemistry, chromatography, and mass spec. I moved over to the cannabis sector probably about half dozen years ago. Initially in 2015, we got our first set of licenses. We were also been involved with running producers as well, and now I'm focused primarily on R&D and cannabis testing here in Canada.
Awesome. Thanks, Rob. It's so great to have you. And I think your perspective from university, I know you've done some work with the National Research Council, and you've got a lot of expertise to bring to the table. So thanks for being here. Kelly, jump on in. Last but certainly not least, good to see you.
Thank you, David. Thanks for hosting. It's great to be here with GMP Collective. My name is Kelly O'Connor. I'm the Business Development Manager for Columbia Laboratories, specifically our cannabis department. Columbia Labs was the first lab to test recreational cannabis in the state of Oregon. Feels like a long time ago now, back about 2017, 2016. We were the first lab to get ISO accreditation for our cannabis testing, and it's been a wild ride. We also test for a lot of different industries, food and feed. We analyze supplements and network, cosmetics, working in several different regulated industries. I like to say that cannabis is the gateway compound entering into a multitude of markets that all have different regulatory parameters. So I'm excited to share our experiences as we continue to help to meet those regulatory parameters through diligent testing.
That's awesome. And yeah, I think your perspective from you're talking about food and dietary supplements and ag side, there's some parallels and there's some lessons learned and uniquenesses. So this is really exciting. So to kick us off, Rob, everyone, I'm sure if you haven't heard about lab testing, I don't believe you. I'll just say that, especially if you're here. But so everybody's heard about, common lab testing problems, from sampling issues, the talk about lab shopping and THC inflation. So I want to start with you, Rob. In your opinion, have we been defining these problems adequately? And, kind of share a little perspective on that.
Yeah, so in the last while, I've been focusing a bit on this issue of in-store testing and the correlation between labs. Everything has been framed in an idea that lab shopping is the responsibility of the labs. It's not. The people who are lab shopping should be equally or more responsible. They're the ones that are making money. But in defense of that, a lot of people have suggested, well, there's no standard methods. There's no inability to consistently test this. And that's completely incorrect. In Canada, we've been running proficiency testing programs for about 4 years now, twice a year. If you look at the results from that, the relative standard deviations are quite low. So it's something that's easy to test. In fact, when you're looking at an analyte that's in percent or the part per thousand region, this is not something that's challenging. We look at things in the natural health product sector or in terms of the US, the dietary supplement sector. These are analytes that are quite readily able to test. If you look at other jurisdictions like Europe and Australia, where they enforce the limits, there's not a THC inflation issue. So it's kind of uniquely a North American one. And it's not just in the US. We found about 30% testing below label claims. But even then, as I think through this, rather than say it's the labs, what I'm coming to the conclusion is that it's not a fit for purpose test. So if you test the harvest at the very beginning, even if you sample and you get a good average value, that will be at best an average value of that harvest. It tells you nothing about the variance within the harvest. And the different growers can have much tighter or more diverse variance within that harvest. So we don't have any information on that. And people are using that high value that they're getting from the harvest and putting it on their labels. It's not fit for purpose for labels. Another thing that's kind of unique about cannabis is as it's processed and it goes through mechanical handling, you're losing some trichomes. And so the variance between buds is actually increasing quite significant. So it can be as much as 30 or 40%, as much as we had 48% in one case from the initial one. So you need to test the finished product. And this is something that is very reasonable. We're able to do this in dietary supplements, and that if you look at a powdered echinacea product, it's not that far off of label claim. If you buy caffeine in a capsule, you're not going to get something that's 40% below that label claim. It's been tested as a finished product. So we can test it. We need to focus on fit for purpose. When we did inter-lab studies, we found that when we sent common samples to other labs, we were within, a couple percentage of each other. So we're able to do the test, but clearly the manufacturers aren't testing at the right point. We have something different in Canada is that we only sell packaged material. So a 1 gram pack is 1 gram. Whereas in some dispensaries in the US, you can basically subsample a bulk. Those are both totally different problems in terms of what the THC content. So we need to focus on what the real issue is here and make sure that we channel and have proper fit for purpose testing.
Thanks, Rob. And I think that you've just identified probably at least 5 different issues that tie in at a high level, and tying it back to fit for purpose, right? It's looking at the devil's in the details, right? With there's a difference between an edible, there's a difference between bulk flower, there's a difference between a pre-roll and packaged flower. And if we're not considering that when we create our scheme for testing and how we get that single number at the end of the day, which is what I think part of the problem is as well, where we're doing ourselves a disservice and kind of glossing over the real issues. Kelly or Jini, I don't know if either one of you want to jump in and add to that with any perspectives or from your side.
Yeah, so when it comes to sampling, so Florida is very strict about their sampling protocol. There's all kinds of requirements, and we have to sample in final product packaging. And then once we take that sample, we then have to composite it before we can run potency. And similar to kind of what Rob was talking about, you can really see the differences even in that packaging. So we actually took 2 separate cultivars, and we took 2 3.5 gram containers that were part of our retention sample, and we dumped all the buds out and we analyzed each of the individual buds. There was about 10 buds in each one. And, in one of those cultivars, we saw a difference of just in those 2 jars, 5.7% difference in potency. So we're talking, 17% to, 23%. That's a drastic jump. And so if you're not sampling properly, you really are going to be skewing these results. And so, sometimes labs are being accused of inflating numbers when maybe it's not the lab. Depending on how the sample came to them, those numbers may have already been manipulated in some way based on the sample that was taken and the sample that was sent to the lab. And so I really do think that fit for purpose, collecting that sample in that final packaging is just essential to providing consumers with the truth and providing consumers with the real numbers and providing consumers with an idea of what they're actually getting.
I love it. Is it fair to make an analogy of I'm envisioning one of my favorite snacks is peanut M&Ms, and imagining that they're all different sizes in that package, and like you test one and you're like, that's 23%, and then that one's 17%. And imagine if that was sugar, right? It's like some M&M packages have way more sugar than others. That we've got to characterize that range, and we're not dealing with M&Ms, we're dealing with a much more heterogeneous product.
Extremely heterogeneous product. Like it's crazy some of the stuff that you can see. And so I do think sampling is a really, really big part of ensuring that your test results are correct.
Mm-hmm. Well, thanks for sharing that perspective. Kelly, I'm sure you've got a few thoughts here on like sampling and other root causes of the situation. Jump in.
Yeah, absolutely. We've seen a lot of variation there. If one day we can blossom into a somewhat self-regulated industry, just imagine some distant future where, companies making cannabis products are able to collect their own materials, send it on in. That's a risk for sure, because as we said, standardization of sampling will help to reduce the variances of the products, help reduce any inflation capabilities as well. But that is the direction we're heading into. And so I would just say, as it stands now, we have regulation handed to us state by state for how to collect samples. In some states, it's required that the laboratory go on site and collect the samples themselves. So that should be an area where there's reduced variance. But it's still an area where we see so many issues, right? And even if the regulations is handed down, even if the labs are supposed to do that type of sampling. So that's why when you're looking for consistency in results, and if you're on this webinar and it's your responsibility to help regulate those practices, seek for the standards, which do exist. There are reference materials available within ASTM and a couple other organizations that can give great guiding practices for, the maximum amount of sample that would still be considered representative. The increments for compositing, as Jini mentioned, that's kind of a very key detail. These are very technical, scientific outlines, right? That in a self-regulated quality system, say if you're working in food or supplements, people have honed in on where they can achieve the most consistency for their product. But again, at this stage in the life of cannabis regulations in the United States, we see that it's often handed down to us through regulatory pathways. So that is kind of my 2 cents on that. I would just piggyback on what Jini and Rob have already said. That is one of the biggest issues of variability, and areas where we can inflate numbers or they can be deflated by certain types of selective sampling. But to provide a appropriate for that product, fit for product, like Rob was mentioning, sampling program that has been validated, meaning that if we sample in the same way over and over and over, we understand what the range of testing is on those results. that'll tighten up anybody's program. This webinar is not for people who are trying to cheat the system. It's for people who are looking to create consistency, to understand more about their product, and to make sure that their testing is actually representative of the product themselves. This is how we can help you with those ways at any of the laboratories here on this call or through the GMP Collective by providing some guidance and standardization of those sampling practices per the product that you're working with specifically.
I love it, Kelly. And I think you've touched on a few important points. And this even goes back to something we were talking about briefly before the show started with Rob around, and it links back to a question that I got or a comment in registration around the cost of testing. But you just point out the value of testing, right? I think we need to reframe this narrative and reframe how we think about it with the value of collecting data to understand your process and what the variability is, and that protects you. It's a lot cheaper to invest in data and sound data collection that you can use to legally defend yourself than to say, well, this one sample must be everything, right? Rob, it looks like you've got, I want to jump in.
Particularly on that thing, because I read it in the questions and it kind of blew my mind a little bit. There are some aspects of testing that are expensive. So pesticide testing and mycotoxin testing can be expensive because the nature of the test. And those tests, ironically, are fine to do on the harvest. Potency in our lab in Canadian dollars, we're charging $80 for potency. This compared to a harvest of 50 to 100 kilograms, that seems like a very small amount. And even if you do a series of 5 or 6 tests on a finished product, that is still not a big number. And if people were held accountable, if the number wasn't right in the finished product and it was independently proven, if that package was recalled, the cost of that compared to the cost of an $80 or even a $400 if you do it multiple times is insignificant. We can do this, right? We do it with other products. And the other thing I just wanted to point out, again, back to this, the lack of methods and consistent methods. In the PT test we have in Canada, the last round, there were 24 different labs involved. One of the products was right around 100 milligrams per gram product, right? The variance on that was 6.5. Now, there is no constant method. People use different extraction solvents, but they do get a powdered material. So sometimes when people send products to different labs, they send buds, and those are not very consistent. So we actually prepare samples for PT testing, and we have to homogenize, we filter, we put it through a sieve. It takes a lot of work to get a homogeneous material to make sure a PT test works, but it's not on the testing thing. And even people get, well, it's the processing, it's the grinding and all of that stuff. We've gotten fine results just with a modified coffee grinder. It works fine. Particularly when you're looking at variance in the products of being that high, we can get reproducible results. So a lot of the arguments, again, are discrediting the very group, the testing labs, that are the ones that can reveal the problem. So that's always been one of my pet peeves.
Good, Kelly.
Yeah, just jumping in and to see it as the point of view, as a producer, as someone who's making these products, right? The R&D has to be available without consequence. This is a huge problem in cannabis right now. If they, for hemp products, for example, if they're testing past a certain threshold in process, is that going to be reported to an authority for them having a product that's considered to be past the actionable level for a certain constituent, such as THC, okay? So if they're working on remediating this and they're trying to get a product into compliance, but their regulation says that every time they test higher than a certain amount, it has to be reported, there's consequences, they might go to jail. This is going to prevent people from wanting to do R&D. We've had to talk to so many companies and let them know the process for reporting in our state for the DEA registration, which I hope to God falls off the hemp bill, for the USDA who's regulating this as well. Everyone wants to know, well, do I need to live in fear for trying to get this right, for trying to get this product to be tested so that I understand more about it? There are certain regulatory frameworks that do crack down on that as a problem. I personally think that this feeds into the issue of lab shopping and potency inflation and all those issues if producers and manufacturers are not able to test for R&D without fear of getting in trouble by the government. So that's an area where I know it's really great in Canada, and that's not really how it works up there, but it's something that a lot of our clients are very concerned about, and we're constantly looking for, reassurances. Now, I'm not saying that the USDA is regulating anything beyond hemp. That's specifically talking about when I'm talking to a farmer that's growing it in the ground,
And just
right?
the plant material, right?
When I'm talking about the FDA, who basically threw their hands up and said, we're not worrying about this anymore. But, those are product relations that I'm talking about. So again, different categories, different regulatory parameters per product, but at every level, people are concerned about this type of consequence for doing their scientific research. So I would just say, that is something that is an ongoing point of education and hopefully development to open up those doors for more science on the plant.
Well, and I think you've hit on a couple things that are important that I want to just recap for everybody listening here. There's a major element of trust, right? Or lack thereof. And I think it pervades throughout the entire marketplace here, where it's easy to point fingers. We've been talking here about how, hey, labs are not the problem. And, hey, we're all labs here. You three have labs here. So let's recognize that, you're naturally going to say, perhaps, hey, I'm not part of the problem, but you actually have the data to show that, right? And the problem is multifaceted. It goes all the way back to the regulations, right? So when lawmakers are creating the regulations, that's a lot of times boxes, different state regulatory agencies that promulgate rulemaking, where they've got frameworks to work within. And it's when you put pesticide limits in regulations, holy crap, we literally need an act of Congress at the state level to change these things. And so that, that gets us stuck. And then you've got producers that, you think about the culture of, being historically an illicit product in marketplace and having trust with government just after decades of prohibition is a boundary that we have to push through and work to and recognize that that's an issue. So then when you bring up your example, Kelly, of if the R&D sampling is not allowed and they could potentially be chastised or, fined or, a negative consequence for a failure, well, they don't want to. It's like, I don't want to ask that question, right? Don't ask a question you don't want the answer to, if it could have negative implications. So we need to change that framework. And, it's a multifaceted process. We're working towards, I love the self-regulation idea where we can work on descheduling, we can work on policy changes. This is all incremental. There's not a, oh, bam, we change it. We've adopted Florida's requirement of, requiring sampling by an independent entity. It's not just that simple, but we've got to start somewhere and start chipping away at these things. And we can show through, self-regulatory frameworks with standardization, again, where they're not in opposition of the current laws or regulations, that we can take the right step and make sound decisions. I just want to touch on a couple of things in the Q&A, and then I'll go to you, Rob. I think, yes, clarifying that USDA does only regulate the hemp plant material, not anything else. That's another major issue. Just a quick question for you, Rob, and then I'll let you jump in on your comment. To confirm, are the PTs blind? I just want to double check on that.
Yeah, the PTs are blind, but recently we're doing a pre-roll study and we sent samples to the 8 labs participating in that, and those are not blinded. So we saw the results for this. Mm-hmm. In the PT, there's roughly between 20, last one was 24, roughly over 20. There's typically 1 or 2 that fail. And when we call a fail, that's a Z score over 2 or below -2. And a Z score is just the number of standard deviations from the mean. So there is only, most labs can get this right. But I do want to clarify, I'm not saying that the labs are not all part of the problem. Mm-hmm. It's labs are able, it's not the fact that they don't have the ability. Labs do have the ability to test if they're ISO accredited or if they're regularly doing PT studies, they're able to do this. There could be some coerced labs that are involved in lab shopping. We can't rule that out, of course.
Yeah.
But to me, it's ironic, particularly in the US, it's like you have some labs that are willing to prostitute themselves, and we use that terminology. They're the ones that get the criminal activity. What about the people who are hiring those people who are processing? They're the ones that are making the money after all. They're the ones that get more money when it's a 29% or a THC as opposed to a 24. So that is, that they should be the one they focus on. The labs should be certainly, there should be a regulatory. They should lose their accreditation if they're not doing it right. But we can do it. It's not that we can't. We do it for other.
Yeah, I completely agree. I think that I appreciate you clarifying and adding that context. So, kind of just building a bit further on, let's talk about THC inflation, which we've kind of touched on a bit. I think you've mentioned things, considerations of the size of the bud, right? And some of the variability you've seen, even Rob. What is the problem? What is problematic? Like, what's the issue there around how labs are performing potency testing? Is there an issue within how the labs are performing the issue and performing the testing? And I think maybe even to the extent that it impacts, Jini, you brought up a point around, how to actually do the subsampling, right, of the product when it's received. Rob, I'll let you go first, and then
so the proper way to do testing, if you're a lab, if you're sent a sample, you have to homogenize the entire sample you're sent, and so that the result that you provide is only valid for the sample you receive. That's point one. But we've also done similar to work with Denny, and we were able to buy 28-gram bags of cannabis, and we broke those down into size. And we found that if we broke it into 4 parts, just roughly on size, there can be 25 to 30% difference between the biggest and the smallest. And even more so, the thing that I think people are missing is as the buds are processed, as they go through the trimming, maybe sorting and other things, there's a lot of tumbling. And when a trichome comes off, that's where the THC content is. And in the very smallest buds, they're not only the smallest popcorn type buds, they're also bud fragments that have undergo extensive contact and physical tumbling. So that's where you get the high variance, and that's why you have to test if the purpose is a label claim, then you have to test the package. But yet we're leading ourselves astray because the people who are buying cannabis, the wholesalers, only care about THC content. In some jurisdictions in Canada, in order to get a high quality bud classification, has to be over 27% THC, total THC. So that's the sum and the correction for the mass difference. That is incredible. Imagine if we were doing wines and we were just, I want to have the highest percent alcohol wine. Nobody would correlate that with quality. And because the governments and regulatory people are so paranoid about the dangers of THC, they've only allowed THC to go on the labels. So then that's become the only thing customers can correlate is THC is related to my quality thing. And that is just, it's nonsense. But because we have that on the label, if you say it's a duck, then you better be a duck, right? So you have to have this honesty. If you say it's a 28% label, and if it comes back at 20%, well, there should be consequences of that. And the latest one, the labs when they test, we have a very tight distribution that is 8% away from where the label claims. So it's not the fact that we're not testing. Well, anyway, sorry.
If we could solve the issue around THC being the sole number at which we define quality, which I think is changing, but as you mentioned, there's a lot of forces working against that. So again, it's not a lab issue per se. It's not a cultivator issue per se. It's cultivators, it's distributors, it's how we tax the product, it's how we approach the product, it's how we regulate the product. It's all of us, right? It's how consumers, consumers being educated. And I like to say we could play the blame game, or we could all just say, how can we solve it, right? What are we doing to be part of the solution? So with that, Jini, I think you would go to you for your thoughts.
Yeah, so kind of what Rob was talking about, sampling is an important part, but also you have to look at where the other error is introduced. And so looking at, sampling is one part of it, but how are you compositing your sample? Because depending on how you're compositing your sample, you can affect that potency number. Depending on how you are calibrating your instrument, what CRMs you're using. We've been hearing recently about people CRM shopping. They buy different CRMs and they see which one is going to give them the best calibration to swing their potency the highest. And that's the CRM they're going to use because it gives them an elevation. And when you look at their data, you'll never be able to see it. Their calibration looks great. So unless there's a way to figure out how to regulate that portion, you still have issues happening there. And, some labs may be using CRMs that they don't realize are skewed. So a lot of this goes into figuring out measurement uncertainty. And I know a lot of labs hate that word, but are very familiar with that word because of ISO. But measurement uncertainty is such an important part because there's measurement uncertainty in every single step of your process. There's measurement uncertainty when you weigh your sample. There's measurement uncertainty when you add your solvent, when you prepare your dilutions, when you run your calibration, when you integrate your peaks. And so all of this stuff creates this compound error that in the end can affect that final result one way or the other. And in some instances, it may deflate the potency, but in other instances, it's going to inflate the potency. And so I think recognizing that as well of, sampling is a big portion of this, but also what the laboratories are doing internally is also a big portion. And the regulations and what rules they're following are kind of affecting all of that as well, because they're doing what they're being told to do, but are they really validating their processes? Are they really determining their measurement uncertainty?
Oh my God. Yep. And just to touch on, and then I want to go to Kelly. When my lab days, I have not ran a cannabis sample in my life, so I'm not going to pretend to know anything about that world besides through you guys. But when I worked in the environmental testing labs, you mentioned CRM shopping. It was understood that there was always going to be some sort of characterization. Like we would actually use, for example, with oil samples, we'd use different 5W30, whether it was Exxon or Shell or Chevron, and the fingerprint looks a little bit different. It still meets within a defined set of, a variance in a range, but it's different. And if you can pick up on that and say, from a nefarious standpoint, it is possible to either knowingly from a nefarious standpoint or unknowingly end up high biasing or low biasing, because what is the CRM to trust and what's the acceptable uncertainty? And that probably even dives into what's the stability of the CRM, right? How is it handled? Chain of custody. And I think that really just brings us back to the measurement uncertainty. You have to characterize all of your unknowns and all of your variances and go back to processing as well, right? Where Rob was talking about how are you handling the harvesting, and the trimming, and is it going through a tumbler, and how are these products actually being fragmented out? If you're not characterizing that as a producer, then that means you don't understand your process and the variance that you're creating into your process. It's just documenting 101 of all of these variables and taking the time to understand it. Go ahead, Rob.
Yeah, just on the CRM, there's a reason we created national bodies to do things like the kilogram. The National Research Council has the Canadian kilogram, NIST has the American kilogram, and everything is balanced against that. So the NRC here in Canada does have a CRM that they have for cannabis material. It's quite expensive, not everybody's using it. But I think NIST is developing one in hemp material because they're tied up with this legality in the US, which, something has to be created, fixed there. But there are things to do that. That's how you get around the CRM issue. But again, I don't want to hit it over and over again, but I will. In this last thing that we did on pre-rolls, okay, I had 8 different labs participating. I sent 6 different pre-rolls from 3 different packages to each of these labs. They each used their own method. And this is before we came with the standard method. The percent RSD on those samples were 6.2%. It wasn't that we're able to test it, but we were 7% against the label claim. So there's no way that there's any inability of the labs to do that. So it's really a false herring. The real thing is making sure the labs get samples that are representative of the label of the package, or whatever. You need to define the problem when you define what's representative. And the second thing is just be honest with it. Get rid of this THC thing. And wine is a good example. Nobody, well, maybe when I was in university, I might have once or twice looked at the percent alcohol when buying wine. But since then, the quality is unrelated to the alcohol content. And we rate the wines differently. If the liquor store only had percent alcohol on the label, we would have a different problem. And the same thing with cigarette. It was only if nicotine was the only thing on the label, it would just get higher and higher and higher. So we've created a problem that we need to correct as well.
Yeah,
Good.
I'm going to jump in if that's cool, David, on that one. Just a couple things to touch on there. You know, labeling and how it ties together with measurement of uncertainty, bringing together what Rob's discussing and what Jini is discussing as well. There's been a lot of issues that we've seen where specifications, whether internal from a manufacturer, whomever, or the specifications for allowable amounts of whatever in a product handed by regulators is not scientifically applicable. This is a huge issue and definitely encourages people to lab shop, either inflate potency numbers or deflate them. For example, I'm going to throw out the example of regulating within 0.5 milligrams per gram for edibles in Oregon, and how tight of a number that is for formulators to hit. It's very difficult for them to achieve that narrow range of specifications because there is inherent variability in testing. So there's a range of acceptable values. A lot of people ask, well, what is a measurement of uncertainty, right? What is it? What's that magic number, right? Well, I'll complicate this a little bit more because it's per product, okay? And it's per laboratory and their proficiency testing, and it's per analyte as well. So if it's around THC, if it's around CBD, those things can have different ranges of acceptable variance. Going a little bit further, measurement of uncertainty is an RPD, relative percent difference, which means that that number is based around the target number. So if you have a target concentration of 10 and the RPD of that is about 10%, you're going to get a range of acceptable numbers between 8 and 12% on that product, right? Keep it pretty simple here. And so if someone gets a 8.5, but their spec was 10, they go, oh my gosh, it's completely out of specifications. In fact, it's not. It's analytically accurate and correct to report those numbers. Had you tested that same exact product a few more times, you might see some numbers higher than that concentration, some numbers a little lower. In fact, you're working with a pretty good lab there that's able to produce within their range consistently around that number. But that range, say it's 8 to 10%, we'll say that the range for the regulations of labeling is that 0.5 milligrams per gram. And depending on the formulation, they might be testing with analytical accuracy, but failing outside of the category for the allowable limits on their label. Now, if that doesn't make you want to like finger pick your analysis numbers, I don't know what else would make you want to. It hasn't been taken into consideration, and it's a big gap in our understanding. So I'm just going to mention that. The other thing that Rob's talking about is THC numbers and the dangers of requiring those label requirements. As he said, it makes it so that that's the only quality parameter for the product. There's a million different examples of different types of products that we could set a standard against. My favorite is comparing hemp to cotton. The USDA has set very clear parameters around what the values are to qualify cotton as a high value, medium value, or low value crop. Has a lot more to do with the additional parameters other than just like, you know, the density of the cotton. And so those are all taken into account to take a profile that's a standard that's set by the national government so that people can test against that. And that's what sets the standard. It's not just one number. It is very dangerous to just list it as THC CBD. To this day, we've only begun to tap into the potential of the plant and the multitudes of constituents. Israelis have discovered more than 160 different cannabinoids in cannabis, and we're only requiring labeling on 2. So talk about a misnomer there. But again, it does create this polarity between those two analytes, which in fact are complementary, and pushes manufacturers and producers into an arena where they feel like they need to showcase their product just based on those two things. We talked a little bit about how trichomes holds a lot of the cannabinoids and different analytes there. There's also flavonoids, there's terpenes, there's a million different markers of quality for cannabis that go way beyond THC and CBD. So I'm going to fully support Rob on that assessment that he's made there, that it is highly dangerous to solely require the testing around THC and CBD. Lastly, I'm going to leave with kind of a cliffhanger there. When you are only testing for CBD and THC, THC is a family, it's a category of different analytes that we can quantify. So now we're looking at any stereoisomer of THC, delta 8, delta 9, delta 10, all the deltas of THC and the variances thereof that are like one molecular bond away from delta 9 THC. So now that we've even more so hyper focused on delta 9 THC, we have left a huge window of opportunity for all these other types of intoxicating cannabinoids to come out into the market and skate by regulations because we're so focused on one particular analyte that we've allowed for room for all these other issues to develop.
So I don't know about you guys in the audience and even Rob and Jini, but it's easy to get overwhelmed by the reality of what you just pointed out, Kelly. And that it's not just you, right? Rob, you've mentioned several things. Jini, you've mentioned several things as well. Suffice it to say, this is complicated and there's no simple solution. And you need to balance innovation with the analytical technology and the ability to actually do it right. I think as an industry, I would argue that we run before we crawl and walk. And again, the framework, Rob, Jini, your labs, Kelly, where I've heard folks getting like CBD socks and tattoos, and it's like, go test this. Well, hold on. This literally shows up at your doorstep at sample intake at 10 a.m. on a Thursday. And it's like, yeah, I'll give you a 5-day turnaround time. You should be developing a method for that modality. We're beyond even just cannabis flower and oil and different matrices. Rob, I know you've given talks around the difference between chocolate and edibles and how that's a completely different matrix. And if you don't perform the correct extraction and validate that, you can get different variables because of the matrix interferences. So this is really complicated. Then let's add in another mix that you just touched on with stereoisomers. And did you know that there's 4 different, I'll call them flavors for simplicity, of delta 9 THC, and that the plant produces 1 in a dominant quantity, but there's 3 others? And who's looking for those? Are you guys testing for those? Do you have CRMs for that? Why, is that a 7-minute method that is a commercially available method for $80, or is that actually more on the forensic side that perhaps should be done during the development phase before you put a product out to market? I'm going to pause there before I go down any more rabbit holes. Rob, what's your thoughts on kind of that overwhelming smorgasbord of this complicated science?
We can make it sound complex, but it's not really. This is not any more complex than other natural health products or botanicals that have been sold for a long time. If you look at European pharmacopoeia, they actually have many medicinal products that are herbals, and they're able to get those things right. And the analysis of those compounds is not more difficult than THC. To get back to the Kelly's statement, but right now, regulatory wise, for international, we have to delta 9 and THCA are the 2 things that we have to measure. We do 17 different cannabinoids, but we're only reporting those for label claims. But we can make this more complicated than it is. We can get this right. They get it right elsewhere. We can do it here. We just have to be mindful. And to get the other thing with Kelly as well, when I say fit for purpose testing on a packaged material, let's say a chocolate or something, there's the label claim, which is when it's produced. But the other issue that's not addressed is the shelf stability, because things will change over time. Ironically, total THC is a little bit more robust because the THCA is what oxidizes the THC, and then the sum stays pretty consistent until you create some CBN. But that CBN conversion is a little bit slower. So that should be measured as well when we're doing potency. But in Canada, they had this bizarre thing that on packaged materials, if you don't do a stability test, you're not allowed to put a shelf life on it. So as if it's infinitely shelf stable, which is insane. We have to take a look at that too. Over 2 years later, you're not going to have the same THC content, CBD content, or CBN content.
And I know I'll give a plug to NRC, as you mentioned, National Research Council of Canada, which I call, folks that are more familiar in the US with NIST, the NIST of the North, National Institute of Standards and Technology. I'm glad you like that one, Kelly. But there's a great paper that they published in 2021 looking at that in flower, looking at stability. And to your point, and thank you for bringing us back to, it can be overwhelming, but this is not impossible. This is not something we've never done before. And to highlight a few, with the remaining 10, 15 minutes here, I want to talk about some of the solutions that exist and give some folks some hope, right? Because in tying back in, I know we won't get to answer all of these questions that have come up, but I'll touch upon them. There is one related to RPD between different labs and, off the shelf, like potency inflation, like what should the acceptable variance be is essentially how I'll read that question. And it comes back to, US pharmacopoeia, European pharmacopoeia, and ASTM have all developed standards around this. And European pharmacopoeia's draft currently suggests 10% variance is the acceptable criteria for cannabinoids. US pharmacopoeia and the ASTM standard suggest 20% variance is acceptable because of the heterogeneous nature. Is one right and the other wrong? It's not about right or wrong. It goes back to fit for purpose. And is there a reason why a tighter criteria of 10% is perhaps needed versus 20%? As long as we're asking the question and putting a number out that the general industry agrees upon, let's introduce the concept of consensus and a process for that already exists. We can do this. We have the answers. And stability studies, right? We have that. That's stability studies, shelf life studies, been around for decades in food, in pharma. This is not a new concept that we have to come up with. No other consumer good that has potential risks because of the shelf life, you don't just turn a blind eye to. That's required to conduct testing. And there's best by dates for a reason. So we can do this, right? We don't have to reinvent the wheel. Rob, go ahead.
Yeah, just to follow up to what you said the 10% in European pharmacopoeia, you're correct. But look at the consequences in Germany. In Germany, if there's a product that's outside of that, the whole product is recalled. It's not, and it has to be destroyed, right? So fundamentally, strangely enough, they're not having an issue with this. In Australia, it's 20%. I kind of lean towards the 20%, but if you're testing in the finished product, so with these pre-rolls, which are not powdered material, there's a big variance between things. It was less than 10% variance between different products. And that's with variance from different labs doing different procedures. It's probably tighter than that, around 5%. So if you say 20 on flower material, it should be fine. So we should be able to do that.
I agree. Jini, jump in. I want to hear some thoughts from you.
And I think too, when you talk about, the RPD variance there for samples, I think you also have to look at the matrix type because different matrices are going to have different homogeneity. So yes, flour, I think 20% is realistic. But when you're talking about a distillate is real homogeneous. Unless they've blended a ton of different distillates together and didn't mix it well, your distillate's going to be the same throughout normally. So if a lab gets 82 and a lab gets 80, those are duplicate numbers. But if a lab is getting 82 and a lab is getting 90, that's a different story. But when you're talking about a 20% RPD in 80%, that's a much larger range. It's going to be a lot more difficult to fail that RPD. And so for some of those higher concentrated products, you have to lower that RPD because that relative percent difference range increases as you increase the concentration. And so I think that's something that needs to be considered when we're talking about label claim. I know when we talk about potency inflation, people like to talk about flour, but I know someone had mentioned in here about the 20% delta 9 variance and, getting cannabinoids of 110 to 120%. We have a funny saying we like to say here where you can't have more than a whole pizza. You can't eat your whole pizza and then magically another slice of pizza shows up. You can't have more than 100%. So if you're testing for terpenes and cannabinoids and flavonoids, you can't have more than 100%. Yes, there is some variance allowable, but we're seeing stuff sometimes on the market of 108%, when you add cannabinoids and terpenes together for distillate products. And it's like, okay, that's a little bit too much of an error that we're seeing there. Like there needs to be tighter parameters, but because those parameters are 20% RPD, you have this range now on those high end of products that allows you to get those numbers that are 90, 95% distillate that may not be realistic of the product.
Mm-hmm. Go ahead, Kelly.
Yeah, it's such a good point, Ginny, really. So our laboratory does not, like if we're testing a product, say an isolate that's 95% pure or higher, and we're using a standard method that has say an 8% RPD on it, we report greater than 98%. For any number we see greater than 98%, because we can't legally defend a claim that says 104%, right? Our lab reports go up in court. So it's important for us. We do offer a high purity analysis. It's a totally different validated method. It uses GCFID. It reduces the measurement of uncertainty down to 2% RPD. So we can claim up to 100% with a high degree of accuracy. This is like not available in other places. It makes it really tough because we've turned down $1 million clients because they're like, we need ours to say 99% every single time. And those specs are, again, as you've mentioned, unrealistic using the standard potency analysis. We do see that competitor labs are, 99% every single time, or, coming out at 104%. These are really important times for people to be checking against that information and that data and questioning hyper consistent results with such high concentrations. That said, we are talking about resources for how to fix some of these issues at this point in the webinar. Last week at Food Safety Summit in Chicago, we talked about cannabis, and somebody said, hey, what can be done if I've noticed that my lab doesn't include any QC data, or that we're always hitting 99%, or maybe a competitor is always hitting it 99% with that laboratory? What do they do? Everybody feels pretty helpless against those experiences, as I understand. Report them to their accrediting body and make them accountable for maintaining their accreditations. Anyone could stand up and say, I have a lab open right now, and you could drop samples off and they could print out a crayon report for you. But without accreditation, that's what you have. Certifying bodies, ANAB is one of them, one of many, but that's who ours is. For ISO, will receive those complaints and will suspend an accreditation and possibly take them off of their accreditation status. So there are pathways for reporting nefarious business practices, both to regulators and to accrediting bodies, that I encourage people to look up. I also encourage people to recognize that the FDA has an adverse event reporting system. So if there are products that you think are unsafe out on the market and people are experiencing adverse events, please look that up and share that as a resource so that we can get more data documenting the dangers of some of these products that are out onto the market. But those are a couple of resources that, ironically, I like to give out to people, how to enforce and encourage more oversight over some of the variable practices.
Well, and Kelly, I appreciate you for bringing that up, because instead of asking the question, is ISO 17025 accreditation enough? It should go back to, are we using these trusted solutions to their fullest extent? And that comes back to the solutions, right? You just mentioned, that's something I learned when we talked just a few weeks ago, that I didn't even think about it. You can reach out to the accreditation body and say, hey, I've got an issue. I'm skeptical. Unless you call them, how are they supposed to know, right? So, think about that. Use the vision. The purpose of these independent accredited processes and certifications is because it has a level of, credibility with these safeguards and these barriers. There are escalation processes to get resolution when mistakes happen, because I don't know, maybe Kelly, maybe somebody in your lab just made an honest mistake, or maybe there's something else going on. You want to know that. And unless somebody tells you, how are you supposed to know? You can't be possibly digging around and proactive around everything. So these resources exist. And that includes going back to standards, right? There is a process for us to create and be able to self-regulate and work with industry, regulators, labs, and consumers to develop what the answer should be. Whether should it be 20%? There's a stakeholder process where ASTM worked very closely, thanks to the US Pharmacopoeia's expert panel, to agree with, yeah, 20% is an acceptable variance for cannabis flower, right? For cannabinoids and sampling, how to conduct representative sampling. Ginny, I want to go to you and then Rob, and then we'll finish up with Kelly. What, maybe Ginny, you're kind of newer to ASTM in our world. I want, anything you can share about that process and how that's been valuable, and any other solutions that you want to share that come to mind.
Yeah, so I mean, ASTM has been great because it is, consensus based. You're having conversations, you're talking about, what is going on, what is the core issue, how can you resolve it? And one of the standards that I'm currently working on right now for ASTM is out of specification and retesting. And I think this is such an important guidance that laboratories need to be provided with, because in this industry, I do fear we have a lot of testing into compliance happening. So people will test something, it'll be out of specification, they rerun it until it's in specification, and then that's the result they report out. Or a product fails and they rerun it until it passes, and then they report that result out. And so really providing laboratories with the guidance of, okay, how can you do this? And when is it okay to retest? And when do you need to retest? And when do you not need to retest? As an analytical lab, you should trust your processes. And if you don't trust your processes, then you need to do investigation, and you need to figure out, what happened? What is your corrective action plan for whatever went wrong? And how do you resolve that problem? And so I think ASTM has really given a pathway for people to be able to contribute to some of those things, and not just analytical methods. Analytical methods are great, but we need to think about operational aspects of it too. So, a quality management system and understanding that labs struggle with that. ISO 17225 doesn't technically require a quality management system. You don't have to have a quality manual per se. You have to have a system, but you don't have to have a manual. Well, if you don't have a manual, how do people know what they need to be doing in the laboratory? And so things like that, it allows you to develop some of those things. And then kind of my final takeaway, is ultimately, if everybody, like Rob said, everybody has the capabilities probably to do things correctly. If you're doing things right, if you're validating your processes, we can all get the same answers. But it comes down to making sure all of the laboratories are choosing that option. We see so many times in industry where they choose the option of, they want the work, they want the business. So they're going to do what they have to do to keep the business. And that's not always the answer, because the consumers are who we should be worried about. Consumer safety and providing correct data to the consumers. That's where the laboratories, that's what we're here for. We're a third-party independent laboratory. And so ultimately for me, that is, my biggest takeaway always is, don't compromise your scientific integrity to keep business, because at the end of the day, whenever it does go to court, if you weren't scientific, if your data is not defensible, you're going to be the ones in trouble, not the people who asked you to do what you did. And so that is the sad reality of it. And so that for me is, my big takeaway as a laboratory is just making sure I keep that scientific integrity, and we're doing things correctly and we're doing things right all of the time.
Thank you so much for that, Ginny. I feel continually encouraged by hearing those perspectives, and yeah, there's an amazing amount of resources that we're creating and that already exists that can make it easier for folks to do the right thing. But like you said, they have to choose to do the right thing, and sometimes that's hard. But that's why we have these checks and balances, and need to continue to work together to strive for that and strive for improvement. Rob, I'd love to hear your kind of final thoughts here in terms of some optimistic, solutions that you've seen, and what's working well, and what's on the horizon that's going to make this problem better.
Sure. I guess one of the points I'll leave you with, we did an in-store study of flower material, and the 1 gram packages were any between 28 and 48% away from label claims. Outrageous, right? However, nobody claimed that these products were lower quality. There's no correlation between THC content and quality. But if you put it on the label, it has to be right. And we have to have something, because that's what the users are buying for, even if they're diluted. If we had a bottle of wine that had 20% on the label and it only had 7% in the bottle of alcohol, well, there would be an issue with that. Why we need to set regulatory things, and regulatory people have to do their job. And not only the people that are producing the products, But the people who are selling the products, the stores that are buying based on THC content and profiting as well, if there is something that's below label claim, they can't just point their finger at somebody else. They have to go be accountable. If I go into London Drugs and I buy a caffeine pill and it's 60 milligrams instead of 100, I would take it back to that store. They give me my money back, and they would hold the manufacturer responsible. And the regulatory governing body would also be all over them. I've tested these and when I reported them, nobody did anything. Well, there's lots of variance in the product. We need to have regulatory people that do their job, and we need then I think the problem will solve itself. And as Jenny said, if they're testing in packages, the labs can get that right. And if they're not consistent, maybe they have to do 5 packages to understand the variance, but they should have a label claim within 20% or 15% of that. Quite easy.
Well said. Yeah, thank you, Rob. And it's defining those specifications and those characteristics, making sure there's accountability in all fronts, right? The labs, the regulators, the producers. And that's, something that we need to, there's room for opportunity there. Let's just say that at the least. Kelly, final thoughts here before we wrap up this evening.
Yeah, just thank you guys so much for this kind of volley of information across the country. I'm in Oregon, Jini's in Florida, Rob's up in Canada. David, who knows, you're probably in hopefully Colorado at home now. But we're all in this together. There are really ethical, quality people that are pushing sound data out and trying to create solutions. ASTM D37 is where I spend my time to help with that. Jini's doing the out of specification report, which I support. I would just say that with the last second I have here to say anything, support the Safe Banking Act, because once people are allowed to sue each other for cannabis problems, I think that'll help to self-regulate quite a lot as well.
I hate that you are probably very correct there. There's a lot of weight to what you just described. So, just final thoughts here as we wrap up, folks. There are some amazing resources. We will be posting more. This will be available recorded for the questions that we did not get to. I will follow up and we will get either a post out or an email in the next couple of days with some further answers. There are a ton of resources. The next 2 months of webinars, I'm really excited to dive further into the labeling issues and potency. So stay tuned for invites there. Everything from stability testing to what's on the label claim. We barely scratched the surface there today. And then if you haven't already registered, ASTM's process is open to all. We will be in Denver in 2 weeks, just less than 2 weeks now. For our consensus committee meeting. We have 10 technical subcommittees. I'm really excited to see most of you here and some folks from the audience attend. And finally, again, there's just great resources. ASTM, AOAC, US Pharmacopoeia, ISO standards, they all exist. We don't have to wait and try to reinvent the wheel and work in our siloed bubbles. We can leverage these century-old processes to make our lives easier and solve these problems by learning from other industries before us. So with that, I appreciate you all, both attendees and participants, for sticking around. This has been so much fun. I look forward to seeing you guys again soon. Have a great evening.
Thank you, David.
Thank you.